Hello there, my name is Claire Bishop, and I'm with Belhaven University's Department of Chemistry, and today I'm going to be presenting a project that we've been working on protein serum albumins and their aggregation behaviors under thermal denaturation. So we're very interested in albumin because of its ability to, in the bloodstream, transport lots of different drugs and metabolites. And we also can see that it forms these very interesting aggregation patterns under certain concentrations of our albumin, as well as in the presence of different buffer salts and high temperatures. And it seems like they are undergoing a conversion from alpha helices structures to a beta sheet that looks a lot like amyloid-related diseases. Protein serum albumin has, in our studies, most interestingly, this binding pocket called cellulose site 1, and it's a place where a drug called warfarin binds. And it has a tryptophan in it, an HSA known as tryptophan 212, but in BSA, which is bovine serum albumin, it is known as tryptophan 214. And it is interesting because in bovine serum albumin, we have two different tryptophans, while in human serum albumin, we have one tryptophan, but this doesn't affect too much with our spectroscopic data, as that tryptophan on the outside of our albumin structures in the bovine serum albumin, BSA, doesn't play a lot of role, while the tryptophan within this warfarin binding pocket is important for characterizing unfolding behaviors under certain thermal denaturation conditions. And we like to use bovine serum albumin more than human serum albumin because it is a lot cheaper, more readily available. So in our laboratory, we've liked to undergo lots of different testing methodology, including differential scanning, calorimetry, circular dichroism, dynamic light scattering, and fluorescence spectroscopy. And each of these are very important for characterizing different aspects of albumin's denaturation. And CD is important for looking at its secondary structure changes. Fluorescence is good for looking at that warfarin binding pocket in specific, because it contains the fluorophore tryptophan 214. Our differential scanning calorimetry is good for just looking at large scale different structural changes, and while our dynamic light scattering looks at the formation of different aggregates. So our DSC was one of the first things that we looked at, and we wanted to see what different concentrations of BSA under different conditions of either pure water or concentration standard PBS, phosphate buffered saline, and what that does to the stability of our BSA under thermal denaturation. And it's been shown that under low concentrations of BSA in pure water that we have a greater stability, as shown by our higher midpoint melting temps. But once we get into higher concentrations of our BSA, it markedly loses stability. But once we go and use PBS, phosphate buffered saline, instead of just pure water, it is a little bit lower in stability than our pure water under low conditions, but it shows a wider range of stability over multiple different concentrations. And that's important because our phosphate buffered saline more closely mimics blood conditions of ion and salt concentration. And so from there, we decided to look at our BSA using fluorescence, looking at that tryptophan 214 in our warfarin binding pocket. And we have a lot of different raw data. And we can see a transition state from a folded to unfolded as it heats up. And so we decided to look at this over multiple different conditions, different concentrations of BSA and PBS in water. And we noticed that most interestingly that in PBS, we start getting these two species rather than water. It looks like we just have one species. And this two species is due to light scattering effects as our albumin forms aggregates in solution. So this is very interesting, in fact. And so we wanted to look at this aggregation because it looks like fluorescence is starting to redshift as well as that light scattering. And this is indicating some aggregations that we'll look at later on with our DLS dynamic light scattering studies, because it looks like we're going from a size of 6 nanometers to greater than 10 nanometers as it's heating up in PBS. And so we're going to be using something called singular value decomposition to look at our raw data so that we can form something called a global fit so that we can better visualize our data. So we're undergoing factor analysis to try to look at how many different unfolding species we have under thermal denaturation, because one of the standard literature standards is that there's two species, just a native and then straight to an unfolded species. However, our data and several other studies seem to suggest that there is a third intermediate state as it is unfolding, as indicated by our numbers here. And we can tell that there's around 3 states here because of a greater amount of our weighted autocorrelations and also a lack of negative values. And so undergoing a global fit allows us to visualize this data in more simple terms. We can turn this into this 3 line right here, which indicates 3 states. These global fits also allow us to look at different enthalpy and transition temperatures of our unfolded, native to intermediate, and then intermediate to unfolded states. We also notice that there is a more pronounced red hump right here in our 100% PBS versus our 0% PBS, aka just pure water. This seems to suggest that there is a more pronounced 3 state under PBS blood conditions. And so here's some more CD raw data of our PBS. So we haven't looked at CD yet, excuse me. And it looks a lot like we're having, in this one right here, a lot of conversion of alpha helices in that primary albumin structure to what looks like beta sheet. We can tell it looks like beta sheet because we can compare it with other literature standards. It very closely mimics other beta sheet formation. So we'll notice that if it was just alpha helices, it would have just that sort of 2 hump look, like with this black line number 1 in our second little figure right here. And we can see part of that being displayed in our figure because it does have alpha helices. However, it also looks like we're converting to an anti-parallel beta sheet formation as indicated by this red number 2. And this looks a lot like our red line right here. And what's also interesting is the fact that this looks so close to other beta amyloid species under the same measuring conditions. And also, we notice a complete lack of disordered structure under this global fit analysis. We can see how this green line right here would indicate that we would have disordered structure and that we would have a positive hump that goes up, but we have no existence of a positive hump. So we're not just having random uncoiling as this protein is heating up. And so we underwent SVDs as well, and we got the same kind of numbers with a 3 state. And we also underwent the same global fit, which also points to the 3 state being more pronounced in that 100% PBS concentration. We also decided to look at our albumin under dynamic light scattering instrumentation. And we use this to look at what it looks like in increasing concentrations as well as increasing temperatures. And you'll notice that our green humps right here indicate that as we increase in temperature and as we increase in concentration, there becomes more and more large particle sizes in solution. We don't see very many small particle sizes at all. And this indicates that we're having more and more aggregates forming in the solution. So this all is very interesting because it sort of shows that we have an amyloid-like beta sheet aggregation of our BSA and HSA, serum albumins in general, and which has little to no random coiling. So it's undergoing beta sheet aggregation. This is very interesting because we can possibly use this for future therapeutics because it hasn't been very well studied yet. We could use these hydrogels for different kinds of drug delivery. And because it's also going through this 3-state process, we can see that it's not undergoing our random coiling 2-state structure. So here's our references, and I'd also like to give a shout out to our other people that have been most involved within this project. We have my advisor and my dad, Dr. Reed Bishop, and he's been one of the people that has led me in research for this past several years, and I'm very thankful for that. And also would like to give a thanks to Isabella Pittman, Emily Skinner, Yadi Satterwhite, and Ryan Williams, who have all either graduated or about to graduate with me. It's been great working with them for the past several years and had a lot of fun on this project. Thank you.